Hey guys, welcome to lesson 9 for our disease unit. We only have one more after this and then the review before you take your test. So hopefully you've been finding these videos helpful, feeling great about it, etc., etc. Okay, so to start, you want to make sure you go and read my updates as much as you can. Got some cool stuff in there. I'm going to use Nearpod again. I did the HPV viral invasion activity in my living room so that I could show you guys what we would be doing if we were in class. But since we're not, that's the way it goes. And then like I said last time, I made the Flipgrid into its own thing on your checklist here. So hopefully you can get to this website and do it after you're done. Oops, that's not the website for Flipgrid. This is the website for Flipgrid. This is the website for the Virus Explorer, so you can pick one and learn a little bit more about a virus. All right, so our viral invasion activity, if you wanted to follow along and like do it on your own. Oops, can't do that now because I'm using the pen. But I put the PDF version here. It's safe, don't worry. It's just for me. And so you just click on it and hopefully you can download it. And then you can print it out. I printed out the host DNA and the viral DNA in different colors so you could see the difference. So that's up to you. If not, it's totally fine. When you go through the Nearpod, you'll be able to see all the steps anyway. Okay. A couple important steps to notice. First of all, you've got to cut the host DNA, and then you're going to connect everything together. Oh no, that's cutting and putting it all together. Then you're gonna cut it at a repeating sequence. So in this case, we cut it at CT, 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 and then we attach the loop. So it's kind of like if we had our host DNA here. Okay, so if this was my host DNA, it was just cut at the repeating sequence. And then the virus just kind of like added its DNA. It's kind of like parallel parking, and I'll use that analogy later on. Okay, so we're going to figure out what does that mean? What does it represent in viral integration, especially when it is all about HeLa, or Henrietta Lacks. So how did HPV get into her cervix cells and reproduce? So the host sequences of DNA, those were Henrietta's cells, her DNA. The viral sequences of DNA represented those from HPV. Your scissors represented the restriction enzyme, and their job is to cut. And tape represented the bonds or the base pairing, because remember, the order matters. The sticky ends are going to be those kind of like, they kind of are like the ends of the strands. So sticky ends are where insertion happens. So where the virus DNA comes in. And then the complete sequences at the end with the host DNA plus the virus DNA equals recombinant. So recombine. Okay, that's how you can break down that word. All right, so at first your host DNA sequence was normal. You cut it at the repeating sequence CT, 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 CT. Why was it important to cut it only in one place? Like if we cut the host DNA in multiple places, then it would probably look something like this. Like let's say that, let's use orange. So let's say that this is my host DNA and I cut it at multiple different places. Like then the virus DNA might do something like this. It might start here and come around, or do something weird like this, and just it's not going to be good. So but if it just cuts it at one place, it was too long. So let's say it just cuts it at one place, then the virus DNA can just loop the loop, boom, and keep going on. All right, there are many kinds of restriction enzyme that cut at specific sequences. Why would it be important to avoid restriction enzymes that cut the host DNA at the replication site? So the key word here is replication site. If you don't have a replication site, then you're not going to be able to copy your DNA. So this is the replication site is kind of a AKA like copy center. And if you can't copy your DNA as a virus, then you can't reproduce. Why might it be important to cut the DNA as closely to the desired gene as possible? So if a different gene is cut, then the host could die, which is bad for the virus. So it doesn't want the host to die. Obviously, it wants the host to live. And so it's just going to try to cut the host DNA as close to the gene as it wants as possible. Okay. Now, one of the reasons that viruses are classified as non-living is because it can't reproduce on its own. So it needs to use the like dividing tools of the host DNA in order to reproduce. How will this new DNA sequence be used? The new DNA sequence will be used to make new viruses and malfunctioning human cells. Okay, so we want new viruses or malfunctioning human cells, aka tumors. All right, HPV genes are classified as E. Classified as E for early genes and L for later genes. What is happening? L for later genes. Okay, so let's talk about these genes. So here up in the top, it shows you the order of where the genes are going. Okay, so we've got, I think it starts, oh yeah, down here is the order. So it goes E6, E7, E1, E2, E4, 5, L2, L1. Okay, so that's kind of how the order goes. So let's see what all these mean. All right, so we've got DNA helicase, which is responsible for unzipping DNA. We've got the transcriptional transmodulator. So that's gonna, since it's transcriptional, it's gonna be responsible for transcription, which remember is DNA to mRNA. We have cytokeratin disruptor, which hurts the structural integrity of the cell so that the newly replicated viruses can escape the host. So we want to pop the bubble so that all the baby viruses can escape. Then we have cell proliferation, which increases the growth of the host cell so it can hold more and more baby viruses. Then we have the transforming protein that binds to p53 that stops apoptosis or death. Then we have the transforming protein that binds to PRB, and that is going to help to make the cell cycle go faster, or mitosis. So it's going to increase the rate of cell division or making new cells. Then we have the major capsid protein, which is on the outside. It's kind of like a turtle shell. And then we have the minor capsid protein, which are kind of like pores in your face that kind of allows the DNA to escape. So why would genes like E1 or E2 be expressed early in the overall process? So E1 is needed for DNA replication and E2 is needed for transcription. So without those, we're not going to be making any proteins that the virus might need. We're not going to be making any new copies. What does helicase allow to happen? It unwinds DNA. Why is cell proliferation encouraged by viral genes? So we want to reproduce in order to survive, which is proliferation is essentially a fancy name for reproduction or making copies of yourself. And then what's the difference between the proteins in L1 and L2? Both of these make the structures which are needed once the DNA is integrated. So as you can see right here, we have the L1 protein and then the L2 proteins, and both of these form the capsid, which is kind of like the shell around the DNA. And so in order to make it, you've got to make the DNA first and then the other protein second. Okay, so we've got to get DNA first and then we can make the rest. All right, so that's DNA invasion of viruses in a nutshell. For our vocabulary, we've got 4 words again. Make sure you're going here and you're practicing, especially now that the test is coming up. And then on the work, we've got how can mutations be passed to future generations? What did we figure out? What is HPV? HPV is human papillomavirus. It's a super common, like 80% of people have it. STD that can lead to cervical cancer in women, of course, because men don't have cervical or cervixes. How does a virus get its DNA into the host cell? Basically, it cuts the DNA and tapes itself in like parallel parking. So I drew a beautiful photo here. If this is the host DNA, we've got the 2 strands here you can see. And then the viral DNA just kind of cuts it here in the middle and then inserts itself like it's been there all the time, like there's nothing to see. Okay, what questions do we have? How does this relate to Ebola? How does this relate to COVID-19? And then write down one more question you've got. And then over here, I've got a fun review question for DNA replication, because that's part of how viruses are affected in our bodies is through DNA replication. That's how they reproduce, just like our cells. So we've got phosphate. I put the phosphates as purple. Over here, all the little circles, the deoxyribose sugars, I made those are the pentagons. And then the hydrogen bonds are what connects the 2 sides. Okay, so in between here, like how it connects in the middle, those are all hydrogen bonds. And then each of the 4 bases, I made adenine pink, guanine orange, cytosine green, and thymine yellow. And then we've got DNA replication. That's what we're looking at. DNA is a double helix or a twisty ladder. Anti-parallel means that the strands are parallel, but one side is upside down. So you can see we have all of our deoxyribose sugars on this side facing this direction, and all our deoxyribose sugars on this side facing this direction. So that's called anti-parallel. So still parallel. Wow, can't spell. Parallel. Nice. All right, so what is the name of the enzyme that unzips DNA? We know that's DNA helicase. All right, guys, that's all I got for today. See you next time.